Hello everyone. In this lecture we are going to discuss about a structure of eukaryotic cell. So the eukaryotic cell includes protist, plant, animal and fungal cell. So even though these are all eukaryotic cell, all these cells are not same, or all these eukaryotic cells are not identical. For example, the major difference between the plant and animal cell is the presence of a cell wall and plastids, and the plant cell consists of a large central vacuole, which actually absent in case of animal cells. So there are some slight differences between these eukaryotic cell. So let us look individual cell organelles in the cytoplasm. So cytoplasm is a semi-fluid like matrix that occupies volume of the cell. It is the main site for all cellular activities to keep the cell alive. Various chemical reactions takes place in this cytoplasm, and when you see the structure of a plant cell, it will look like this. And this is the animal cell. Okay. Let's see what is a cell membrane. Cell membrane is the protective layer, or it is a semi-permeable layer which is present around the cell. Even though it is a plant cell or it's an animal cell, it is a bacterial cell, the presence of cell membrane is crucial for every type of cell, as it acts as a semi-permeable membrane. So in case of plant cell, you can see the cell membrane here, which is present all over the cell like this. And in the case of animal cell also, there is the presence of cell membrane. So in the cell membrane, if you see the composition of the cell membrane, in the cell membrane lipids are arranged in bilayers, and lipids are arranged within the membrane with the polar head towards the outer side and the hydrophobic tails towards the inner part. So if you observe the structure of a cell membrane, so the lipid bilayers actually arranged in this manner in the plasma membrane, in which the polar heads towards the faces outside, whereas hydrophobic end faces inside. So this is the bilayer arrangement of lipids inside the plasma membrane. So lipids are arranged within the membrane within the polar head towards the outer and hydrophobic tails towards the inner part. So the lipid component of the membrane mainly consists of phosphoglycerides. So along with lipids, the cell membrane also contains proteins and carbohydrate ratio, which is varies from cell to cell. So that's why due to the presence of proteins and lipids in the cell membrane, the cell membrane is known as a lipoproteinaceous layer. Lipoproteinaceous layer. So the lipid and protein composition and carbohydrate composition in the cell membrane varies depends upon the type of cell. So the composition will not be same for each and every type of cell in the body. So if you take human RBC, the human RBC consists of a cell membrane which have the protein of 52% and 40% will be the lipids. And the 52%, among the 52% of the proteins, there are integral proteins and peripheral proteins. So the most accepted model of cell membrane is the fluid mosaic model, which was proposed by Singer and Nicholson in 1972. And so the quasi-fluid nature of lipids enable the lateral movements of proteins within overall bilayer. So the ability to move within the membrane is measured as its fluidity. So the proteins inside the cell membrane have the ability to move, and the fluid nature of the membrane is also important for the functions like the cell growth, formation of intercellular junctions, secretions, and endocytosis. So in the proteins which are present inside the plasma membrane actually serve the purpose of transportation, and also due to the presence of the cell membrane could be able to form some intercellular connections by forming intercellular junctions, and also the secretion of some products inside the cell or the proteins inside the cell will be possible by the presence of or the fluid nature of the cell membrane, and the engulfing of certain substances will be possible due to this fluid nature, and this process is known as endocytosis. And cell division, if this parental cell have to divide into two daughter cells, the cell must be divided, and the cell division is possible if the cell membrane has this fluid nature, so that the cell could be easily divided or separated into two daughter cells. So many molecules can move briefly across the membrane without any requirement of energy, and this mode of transport is known as passive transport. So the cell membrane do contributes for the function of transportation. transportation. So there are some movement of molecules which actually occurs across the membrane without the need of any energy or requirement of energy. That mode of transport is known as a passive transport. And the transportation process which required energy is known as active transport. Neutral solutes may move across the membrane by the process of simple diffusion along the concentration gradient, that is from higher concentration to lower concentration. So this simple diffusion also can take place effortlessly. It is simply the movement of molecules from higher concentration to lower concentration. It also depends upon the concentration gradient. And the movement of water by diffusion is known as osmosis, and there are some substances which need to be carried by the proteins in the cell membrane, so that kind of transportation by carrier proteins is called facilitated diffusion. A few ions or molecules are transported across the membrane against their concentration gradient, that is low to high concentration, which actually requires ATP, is known as active transport. The example will be the sodium potassium pump in the body. So this is about the cell membrane, which we can see common in both plant and animal cell. Cell wall. So cell wall is a layer which is present in the plant cell. So it actually protects the cell from mechanical damage and infection. It gives a film, a strong shape to the plant cell, and it also helps in the cell to cell interaction. So when you take the examples in case of algae, the cell wall composition is with cellulose, hemicellulose, pectins and proteins. And the cell wall of a young plant is known as a primary cell wall, is capable of growth which gradually diminishes as the cell matures, and secondary wall will be formed on the inner side of the cell. So the middle lamella is a layer which is mainly of calcium pectate, which holds or glues the different neighboring cells together. Endomembrane system is of about the network of these organelles, which is endoplasmic reticulum, Golgi, lysosomes and vacuoles. So let's see these organelles. Endoplasmic reticulum is nothing but it is a network or reticulum of tiny tubular structures scattered in the cytoplasm is called endoplasmic reticulum. So when you see the structure, it will look like this. So it forms a network of tubules in the cytoplasm. So the ER divides the intercellular space into two distinct compartments. One is luminal, another one is extraluminal. There are two types of endoplasmic reticulum. Rough endoplasmic reticulum, which is actually observed in the cells which actually participates in protein synthesis. So when you observe, these are the ribosomal attachment to the surface of the endoplasmic reticulum, which converts this portion of ER into rough endoplasmic reticulum, and this concentration will be high. This number of rough ER will be high in the cells which actively participates in the protein synthesis and secretion. They are extensive and continuous with the outer membrane of the nucleus. So there are another kind of membrane which is devoid of ribosomes is known as soft smooth endoplasmic reticulum. So if you observe here, there are no attachment of ribosomes on the surface of the ER, which develops into a smooth ER. So in animals, it serves the purpose of synthesis of lipids. In animal cells, steroidal hormones are synthesized in smooth endoplasmic reticulum. Next organelle will be the Golgi, which was discovered first observed by Camillo Golgi in 1898. These are densely stained reticular structures near the nucleus, consists of many flat disc-shaped sacs or cisternae of 0.5 μm to 1.0 μm in diameter. So if you observe the structure, they actually arrange, these are like tubule like structures which actually arranged in stack, and it has two phases, cis and trans, and a former phase and a maturation phase. So these are stacked parallel to each other, concentrically arranged near the nucleus with distinctive convex cis or forming phase. This will be the convex cis known as a former phase, and a concave trans or maturing phase. So this is where the newly formed Golgi will be mature at this end, and at former phase the new Golgi will be formed. So the main function of the Golgi is the packaging material to be delivered either to the intracellular targets or secreted out of the cell. So when you see the procedure in ER, the proteins will be synthesized, the newly formed proteins will be synthesized here, and thus synthesized protein will be transported to the Golgi complex to the cis end. After packaging, it will be transported to the trans end. From the trans end, the protein either will be secreted outside the cell, or it may be secreted in some other intracellular spaces. And it also serves the purpose of glycoproteins and glycolipids, formation of glycoproteins and glycolipids. And some other kind of organelles are lysosomes. These are again the membrane bound vesicles. These structures are formed by the process of packaging in the Golgi apparatus, and the most important point one has to observe here, the presence of hydrolytic enzymes inside the lysosomes. Inside the lysosomes, yeah. So these are the lipases, proteases, and carbohydrates. These lysosomes are optimally active at acidic pH. So due to the presence of these lysosomes, so the lysosomes are rich in the cells which actively involved in the phagocytosis process. Phagocytosis. So when you observe the immune cells like macrophages, they actively involve in the phagocytosis. So we can say The lysosomes will be high in number in case of cells like a macrophage. So whenever the macrophages engulf the pathogen, macrophages engulf the pathogen. See, these are the pathogens. So whenever this macrophage engulfs the pathogen, so there are lysosomes inside the macrophages which consists of hydrolytic enzymes. So by the action of this hydrolytic enzymes in the lysosomes, the pathogen will be fragmented into antigenic peptides. So the lysosomes are rich in hydrolytic enzymes, and when you observe the shape, these are vesicle-like structures, consists of granules, hydrolytic enzyme, and some undigested fats. So these are also known as suicidal bags of the cell. So next will be the vacuoles. These are membrane-bound space found in the cytoplasm. So they will be vacuoles are vacant places which we can found in large number in case of plant cell, and in case of animal cells these are less in number. So the vacuole consists of a water slab, excretory products and other materials which are not useful for the cell. It is covered 90% of volume of the cell, is surrounded by a membrane called as tonoplast. So this tonoplast have the application to serve for the transportation of ions against the concentration gradient. So the concentration is high in vacuole when compared to the cytoplasm. So the vacuoles, when you see the vacuoles in other organisms like amoeba, it consists of a contractile vacuole which is important for the secretion, and in case of protist, it consists of a food vacuole, it formed by the engulfing food particles. So the mitochondria, which is the powerhouse of the cell, it is a sausage shaped organelle, or it has a cylindrical shape having a diameter of 0.2 to 1.0 mm, a length of 1.0 to 4.1 mm. It is double membrane. It consists of an outer membrane as well as an inner membrane. So outer membrane forms the continuous limiting boundary of the organelle, and inner membrane forms the number of infoldings called cristae, which increases the surface area. So when you see the structure, you can observe. So this is the outer membrane which is smooth without any foldings. So when you see the inner membrane structure, it actually infolds to increase the surface area, and these infoldings are known as cristae. And the mitochondria consists of ribosomes and small DNA molecules, matrix. So the mitochondria serve the purpose of aerobic respiration. These are sites for the aerobic respiration, produces cellular energy in the form of ATP, hence it is called as a powerhouse of the cell. So the matrix which is present in the inner membrane possesses single circular DNA molecule, few RNA molecules and ribosomes which are 70S, and components required for synthesis of proteins. So the mitochondria divides by fission. And the plastids. So this is one differentiation which we can differ between a plant and animal cell. The plant cell and euglenoid contains this plastids, where the plastids are absent in case of an animal cell. So the plastids are of various types: chloroplast, chromoplast and leucoplast. Chloroplast consists of chlorophyll and carotenoids, and chromoplast consists of fat soluble carotenoid. Pigment like carotene, xanthophyll are present, and leucoplast consists of leucoplast are colorless plastids of varied shape and sizes stored nutrients. So there are some other kind of plastids as well, which are known as amyloplast, which stores carbohydrates, example potato. Mm, Aleuroplast store proteins, and Lalloplast stores oils and fats. So these are wide variety of plastids which we can found in plants. So the major portion of plastid which we have to discuss is about the chloroplast, because the chlorophyll is the pigment which serve the purpose of photosynthesis in plants. So the chloroplast, these majority are seen in the mesophyll cells of the leaves. These are lens-shaped, oval and spherical discoid shape, even ribbon-like organelles having the variable length. So green algae consists of 20 to 40 chloroplasts per cell in the mesophyll. These are also double membraned, consists of inner membrane, outer membrane, and inner membrane is relatively less permeable. The space limited by the inner membrane of the chloroplast is called stroma, where the dark reaction takes place. A number of organized flattened membranous sacs called the thylakoids are present. So in the thylakoids are present in the stroma, and the chlorophyll pigments are present in the thylakoids, and the stacked arrangement of the thylakoids are called as grana. The stroma lamella are the structures which connects the thylakoids of the different grana. So the stroma consists of enzymes required for the synthesis of proteins and carbohydrates, and the chloroplast also contains small double-stranded circular DNA molecule and ribosome smaller than the cytoplasmic ribosomes. So when you see the structure, it looks like this. So these are the outer and inner membranes of the chloroplast. So these are the stroma lamellae, stroma lamellae, and this coin-like arrangement of thylakoids is known as a granum. Thylakoids outer and inner membrane. So these thylakoids, inside the thylakoids we can find the chlorophyll pigment which actually traps the light energy and converts into chemical energy and serve the purpose of photosynthesis in plants. So other kind of organelles are ribosomes, are discovered by George Palade in 1953. These are granular structure composed of ribonucleic acid and proteins and are not surrounded by any membrane. So there are two kinds of ribosomes, is ATS types and 70S type of ribosomes. So ATS type have the eukaryotic cell consists of ATS type of ribosomes. The subunits are 60S and 30S. The S stands for sedimentation coefficient, which is a measure of density and size. So cytoskeleton. Cytoskeleton is an elaborate network of filamentous proteinaceous structure present in the cytoplasm is collectively referred to as a cytoskeleton. It serves the purpose of mechanical support, motility, maintenance of the shape of the cell. So you can see the structure here. It consists of plasma membrane, peripheral microtubules, interdouble bridge, central microtubule, radial spokes and central sheath. So cilia and flagella.